Okay, welcome to Let's Get to the Marks, going through an AQA Biology A-Level Respiration question pack from the physicsandmathtutor.com website. Brilliant website. You need it if you want to get an A or an equivalent website. This one's free. You just got to put up with some adverts. But like me, you can just hit the download to your computer and then you don't even have the adverts. So you get the PDF downloaded to your computer. It's brilliant. It's got the mark scheme, but I'm going to go through with an explanation. So let's get into it. Okay, so I'm going to describe the process of glycolysis so that we can answer this. So if you check out glucose in the cytoplasm of a cell turns into hexose bisphosphate or biphosphate, or however you want to say it. And it uses 2 ATP to do that. So we hydrolyze 2 ATP molecules and that produces these phosphates. Obviously, we're going to get two inorganic phosphates. They must be added to the glucose molecule to make it into this hexose bisphosphate. And that means we can call this the phosphorylation of glucose. So during glycolysis, glucose is phosphorylated. Let's look at the next stage of glycolysis. So in the next stage of glycolysis, this hexose bisphosphate is pretty unstable and so breaks down to form two triose phosphate molecules, each one with 3 carbons. So there's still a total of 6 carbon atoms knocking around here, and that's important. However, it's important to note that this doesn't use or produce any ATP. So in a way, if you were revising, just knowing that glucose ends up as TP, and during the process it's phosphorylated using 2 ATP, that would be the key. But if you want to go that extra mile, you can learn that the intermediate molecule that's unstable is hexose bisphosphate. Okay, now the next important stage of glycolysis, we're going to take these TP, these triose phosphate, and we are going to oxidize them. Now, how do we oxidize them? Well, the TP is going to be oxidized, and it's going to do this by reducing NAD to reduce NAD. Now, we can call reduced NAD NADH shorthand just for ease of communication and for drawing on diagrams. And we're going to need 2 lots of NAD, one for each of the TP molecules, and that's going to produce 2 lots of NADH. And during this process, the triose phosphate is oxidized, so it loses electrons and hydrogen. Now, this process is really important because this process actually produces 4 lots of ATP, and it does this by joining together 4 ADP with 4 inorganic phosphates. Well, I put 4 times ADP plus that. I don't need that last 4, and it forms 4 ATP. So we have a net production overall. If we look at the first stage, the phosphorylation of glucose, we used 2 ATP. We produced 4 ATP in the final stage of glycolysis. So there is a net production of 2 ATP and 2 NADH or reduced NAD. That shouldn't be in lowercase. You would actually lose marks for that. Now, what is this process called? Well, you do need to know this process as well. This is known as the oxidation of TP. So the oxidation of triphosphate. So technically, glycolysis is made up of 2 stages: phosphorylation of glucose and the oxidation of TP into pyruvate. And I haven't even shown that it becomes pyruvate. What a wally. So this, once you oxidize the TP, it becomes pyruvate. And obviously, one glucose molecule makes 2 molecules of pyruvate. So now we just construct that into an answer. And what do I recommend? How do you learn these complex content, these stages like this? A-level biology. If you want to get an A or an A star in A-level biology, you need to learn these off by heart. You should be able to, when you're chilling out, watching Netflix, watching a movie, just chilling in your bedroom, whatever you're doing, you should be able to grab some scrap paper. You should be able to scribble out glycolysis with all these stages, show where the ATP is used, where the phosphorylation occurs, show the net production of ATP, show the oxidation of TP to pyruvate. You should be able to write that all out. And you should be able to do this for the link reaction, the Krebs cycle, and oxidative phosphorylation. And if you haven't learned these off by heart by now, what are you doing? Get on with it. Okay, that's going to get you 4 marks. You can see the answer there. Let's move on to the next part of this pack. Part B, malonate inhibits the reaction in the Krebs cycle. Explain why malonate would decrease the uptake of oxygen in a respiring cell. So you really need to know the Krebs cycle here. So I'm going to draw that out for you, and then we're going to see how it links to oxygen uptake. Okay, so here is an incredibly crusty drawing of the Krebs cycle. You see acetyl-CoA joining here with this oxaloacetate, this 4 carbon compound. You don't need to know the name of that. And it becomes citrate. You don't need to know the name of that. But essentially a 4 carbon compound joins with acetyl-CoA and becomes acetyl-CoA, by the way, is a 2 carbon compound. So when it joins with the 4C, it forms a 6 carbon compound. This then breaks down to a 5 carbon compound. And it's during the breakdown from the 6 carbon to 5 carbon and the 5 to 4, that the really important things in the Krebs cycle happen, and that is the production of NADH, which is the electron and hydrogen ion carrier. And we can see here in this second stage, well, we get three NADH molecules made, one in the first stage from 6 carbon to 5, and one from two from 5 carbon to 4. They're very, very important. We also get an FADH, which does the same job as NADH, formed as well. So they're really important. Why are they so important? So this is the thing. If you break the Krebs cycle, you're not going to get any of this NADH or FADH. Why is that vital? Well, in the final stage of aerobic respiration, which is a super important stage where all the ATP, or 99% of the ATP, is made, we're going to have the electron transport chain. So you probably remember the ETC. Well, it's the breakdown of, say, NADH or FADH that releases hydrogen ions and electrons, right? And those electrons, they supply the electron transport chain. Without these electrons, there is no electron transport chain. And if you have no electron transport chain, you have no need for oxygen as the final electron acceptor. And the oxygen joins with the hydrogen ions to become water, which is a waste product of respiration. But so the keys for this question are that if you break the Krebs cycle over here, then we have no NADH, or you can say less NADH, less FADH, which means less electrons to the electron transport chain. There's no need for oxygen as the final electron acceptor, and so you will have a reduced uptake of oxygen. You can use the word no, so I'm going to say no FADH or NADH. Make sure you write in capitals for those. I wouldn't get marks for what I've done there. So no FADH or NADH produced in Krebs cycle. So no electron transport chain. And then we can say no need for oxygen, which I can't spell, oxygen as the final electron acceptor. Now, realistically, you do make some NADH. You do make some NADH in other parts of respiration, just a little bit. Remember, we made some NADH during glycolysis, and so some NADH would be produced, which could then come on and be used in the ETC. However, I think if you say no FADH or no NADH, it will be on the mark scheme, or less FADH or less NADH produced, which means less electrons for the electron transport chain in oxidative phosphorylation, which means we don't need oxygen as the final electron acceptor, which means the uptake of oxygen is decreased, and that will get you your marks. Excuse my crusty GCSE D grade drawings. Let's move on to the next question. Question 2. In muscles, pyruvate is converted to lactate during prolonged exercise. Explain why converting pyruvate to lactate allows the continued production of ATP by anaerobic respiration. Well, anaerobic respiration is made up of two parts. It's made up of the glycolysis, and I'm not going to draw the whole thing out again, but I am going to draw the final bit of glycolysis out once again. So in muscles, pyruvate is converted to lactate during prolonged exercise. Explain why converting pyruvate to lactate allows the continued production of ATP by anaerobic respiration. If we look here in the final stage of glycolysis, which is part of anaerobic respiration and aerobic respiration, but we're going to deal with the part in anaerobic, the triose phosphate is oxidized to pyruvate. And during that process, it makes a load of ATP. It makes 4 ATP molecules. And this is good enough to produce that little bit of extra energy during prolonged or intense exercise that you might need to perform that intense activity. So we're making that ATP. However, we cannot make that ATP anaerobically unless we have a lovely supply of NAD. The problem is sooner or later we're going to run out of all the NAD in the muscles, and we're going to be left just with NADH, and we won't be able to convert any more triose phosphate into pyruvate and produce ATP. So we need to then take the pyruvate, and what we need to do is get this pyruvate, we turn it into lactic acid. You know that stuff that makes your muscles sore. You can call it lactate or lactic acid. And during that production, the whole purpose of the production of lactic acid is to take NAD reduced or reduced NAD and turn it back into NAD. Why? That is needed back over here so we can have the continued production of ATP by the oxidation of triose phosphate. If we don't regenerate the NAD by converting pyruvate to lactate, we won't be able to convert any more triose phosphate into pyruvate, and you won't be able to anaerobically respire anymore. So regenerates, which I can't spell, so regenerates the NAD, or it oxidizes NADH to NAD. So I could say oxidizes NADH to NAD. Remember, in AQA and OCR, they like to use the words reduced NAD or oxidized NAD. So there we go. Right. So it oxidizes the NADH to NAD. This allows glycolysis to continue. So it's the glycolysis that produces the ATP. The pyruvate to lactate produces no ATP. The formation of lactic acid does not produce any ATP in your body. It is simply for regenerating the NAD to allow glycolysis to continue. If you want to be more specific, you can say to allow triose phosphate, TP, to be oxidized and produce ATP. So, which is essentially glycolysis. And that will get you 2 marks. Now I'm going to do this respiration walkthrough in 2 parts. So if you like the first part and you found it useful, hit the like, leave a comment, and then I'll make the second part to the video, which will cover the rest of this revision pack from Physics and Maths Tutor. Okay, question 3. Yeast cells can respire aerobically or anaerobically. A student used the apparatus shown in figure 1 to measure the rate of respiration in yeast. She positioned the flask in a water bath so that the yeast culture reached a constant temperature, then left the apparatus for 1 hour before starting her investigation. Here we can see we've got a glass tubing, a delivery tube or a capillary tube. We've got a scale. We've got some colored liquid, normally red, and we've got yeast culture in glucose in a little container here. And then we've got this alkaline pyrogallol to absorb oxygen. So this is forcing the yeast to do what? It's forcing them to anaerobically respire, because obviously that's what we actually want to look at. So let's go down here. Suggests one reason why it's important that the student left the apparatus for 1 hour after the yeast culture reached a constant temp. So normally we have to leave them 5 minutes to equilibrate to get to the right temperature that we set the conical flask at or the environment at. However, they've left it for a full hour. Why is that? What's present in the flask that we don't want if we want to investigate anaerobic respiration? Well, the problem is we've got oxygen in the atmosphere here inside the flask. We need that to get absorbed by the pyrogallol and remove the oxygen, which will force the yeast to only anaerobically respire. So that's why we leave it for an hour. So we want to say to remove all the oxygen to ensure yeast respire anaerobically. Awesome. And that will get you a mark. Now, let's look at the next part to this question. So during the investigation, or during her investigation, sorry, the colored liquid moved to the right. Explain why it moved to the right. So I'm going to go up and explain why this moves to the right. So the colored liquid will stay exactly in the middle there if the pressure does not change on either side of it. Sometimes in these questions, they have a second control flask over here, where instead of yeast, they will have the same mass or the same volume of a non-living material like glass beads or something like that. And they'll have that over here in a control. The colored liquid will move towards whichever flask has a decrease in pressure. So we don't have another flask here, but what's going to happen to the pressure inside this tube? Well, if you think about pressure, pressure is equal to force over area. You did that in GCSE physics. And pressure is created by the collision of gas particles with the walls of the container and with themselves, the force over a particular area. What's going to happen to the pressure in this flask? What are the yeast doing? What are they releasing? Well, the yeast are releasing a gas, and that gas is carbon dioxide. Means we're going to have an increase in particles in the flask. They're going to collide with the walls of the container, and that is going to increase the pressure. And when the pressure is higher here, it forces the liquid to the right. Okay, so we would expect the liquid to move to the right because there is an increase in pressure in the flask caused by the yeast releasing carbon dioxide. So you can make sure you have a go at these questions by pausing the video and then writing down the answer afterwards. So increase in pressure um, in the flask due to carbon dioxide release um, pushes liquid to the right. So it's moving down a pressure gradient towards the lower pressure side, which will be on the right-hand side. And that will get us to marks. Look, all those lines, you don't really need all that space. Okay, let's move on to part C of this question. Oh, we got a little maths, biology maths. Wunderbar. The student found the colored liquid moved 1.5 centimeter cubed in 24 hours. The diameter of the lumen hole of the capillary tubing was 1 millimeter. The volume of the capillary tubing is given by pi r squared times the length, where pi is 3.14 and the length, L equals length. Calculate the volume of gas produced in centimeter cube per hour. So pause the video, have a go, and then watch the walkthrough. Okay, here we go. Look, straight away, we've got centimeter cubed hour minus. What does that mean? That means that they have taken centimeter cube, which is the volume, and they've divided the volume by the time. That's what to the -1 means, centimeter cubed over time. So straight away, you need to find the volume, and we've been given the equation for finding the volume. It's pi r squared times L. So we need to do that and then divide it by the hours. And they've given us the hours. So we're really looking at something over 24 hours. We just need to find the centimeter cubed and divide it by 24 hours. So let's get down to it. We're going to need to do 3.14, which is pi. It says to assume it's 3.14. We need to times it by the radius squared. So what's the radius? Right, the diameter is in 1 millimeter. We've got a little trick here. We've been given centimeters and millimeters. Well, look, our answer's in centimeter cubed. So we need to convert that into millimeters. Now, there are 10 millimeters in a centimeter, so we need to do 1 over 10 as our radius. However, 1 over 10 1 over 10 is 0.1, but that is the diameter. So 0.1 or 1 over 10 equals d, the diameter. We need to do the radius. So remember, d divided by 2 equals the radius. So I need to do 0.1 over 2. That's 0.05. And we need to square that, because it says it's squared there, and then times it by the length, which is 1.5, and then that gives us the centimeter cubed. So let's smash that all into the calculator and then divide that by 24. Divide the centimeter cubed by 24. Pedantic. And we're going to get 0.0004906. You could round that up to 491 if you like, because the 6 is bigger than the 0 there. And we could say, we could put it in standard form. So 4.91 times 10 to the -4. It doesn't ask for standard form, so you can probably get away with that as well. And that is going to get you your 2 marks. Beautiful. Right, let's zing on. Okay, figure 2 shows a typical growth population curve for yeast under laboratory conditions. Here's figure 2. We've got log 10, the number of cells, and then we've got the curve going up. We've got the death phase. That looks like the stationary phase and the growth phase or exponential phase. Explain why a log scale is used to record the number of cells. So why do they use a log here? The answer is that here we might be starting with 1,000 yeast, right down here. By the time we get up here, we might be on 100 billion yeast. So if you tried to show that in normal numbers, you'd need a graph that's about 7 meters long on your A4 paper. So that'd be impossible. So we have to use log 10 because it's great for dealing with large increases in numbers. So because we have a rapid or large increase, a rapid and large increase in the population or numbers. Yeah, so there's a massive increase in numbers. You just wouldn't be able to have a graph where you go from 1,000 to a trillion or hundreds of billions or whatever on one side of the axis. You couldn't do it unless you use log 10. So that is the reason why we use log 10, people. You'll often see them on graphs of bacteria or other microorganisms, fungi, whatever. Right. Many yeast cells die during the death phase. Suggests why. So why is it that the graph started to decrease? What is happening to the yeast? Well, What do they use? If you go back up to the diagram, this is where you'd search the paper and you'd be like, oh, why are they dying? Why are they dying? Go right back up here. The yeast are living on glucose. What's going to happen over time to the amount of glucose that we have in the flask? Well, the glucose is going to go down and the yeast are living off the glucose. It's their substrate. It's what they're using for respiration. And so we would say decrease in glucose, less substrate, less respiration. So they can't do respiration, so they're dying. The other things are, what else did the yeast produce? What was that gas? So we could say carbon dioxide is acidic, decreases pH. What's that going to lead to? Denature of um, yeast enzymes. So the enzymes within the microorganism within the yeast are going to denature once a certain level of carbon dioxide is produced. Also, ethanol, I think it's about 50%, which is why they have to ferment, like they have to distill alcohol to make things like whiskey and vodka that are like 40% alcohol. I think the maximum strength alcohol you can get from yeast without distilling it is about 10, 12%. Once the ethanol gets to about that level, it starts to kill the yeast. So ethanol is toxic for the yeast. So it's like they produce ethanol, but then the ethanol kills them. Grimy, eh? Right. Um, cool. Let's zing down. And then the final part to this question is another dirty maths question. Let me shrink that a little bit so that we can get this all on the page. F, the following equation can be used to make predictions of the growth in the population of yeast cells under ideal laboratory conditions. Xt equals xo over E to the power of RT. Xt equals the population after a certain time. Xo equals the population at the start. E is 2.72 base of natural logarithm. Now you actually have an E button on your calculator. You'll be able to find it on your calculator. You sometimes have to press Shift or Alpha to access that E button. So I suggest you find that. If you don't know how to do that, you need to spend some time, Google on the internet, look up your modeler calculator and find how you can access the E button. And if you press Shift E and equals, it should come up 2.7198 or something like that. It's almost 2.72, so you can just use that button. R equals the growth rate and T is the time period over which R applies. A population of 2,000 yeast cells was left for 10 hours. The value for the growth rate was 0.5. Assuming no yeast cells died, calculate the predicted size of the population after 10 hours, show you're working. It's literally just plug the numbers in. So XT is what our final answer is going to be. And all we need to do is XO, and that is the population at the start. So that's 2,000. So we need to smash 2,000 into the calculator. You can put this all in one go in your calculator, or you can separate it, times E. So you'll find there's an E button on your calculator. You sometimes have to press Shift E or Alpha E, or there'll be a button. You'll need to work that out for your calculator and know before your biology exam how to find that exponential button. And then you need to press that, and then you just type in the exponents, the other bits, which is 0.5 for the rate, times T. What was T? 10. So you could just put 5 because 0.5 times 10. So I'd just simplify that to 2,000 times E to the 5. And that is going to get you, let me just smash that in my calculator and I'll tell you what that gets me. So where is my button? There we go. Bash, bash, bosh. Give me 29,700. So 29,766. And there we go. Smash up the bell, punch up the like, subscribe. Let's get to the marks. Wunderbar.
You're still here? It's over.